Well, in today's lesson, we're going to be talking about diffusion and osmosis. What are the similarities and differences between the two processes? And so let's get started. Diffusion really is just all about the particles, just as osmosis is too, but diffusion in particular is really talking about how molecules throughout a particular matrix will move from a higher concentration to a lower concentration. So let's take a look at this picture that I've included. And you've probably seen this demonstration in other science classes, where you put a drop of dye into water. And before we talk about that, a solution is really a mixture of two things: a solvent, which something's dissolved into, like water, and a solute. And the solute is the thing that's dissolved in, let's say, the water, like in this particular case, like a dye. For this demonstration, or this picture anyway, dye has been put in here, and these particles, these red particles, are the dye particles. And over time, what's going to happen is these particles are going to start moving outward and diffuse themselves throughout this matrix of water, throughout the solvent. And as time goes on, they're just going to get further and further apart, like we see here in this second image. And they're going to assort themselves and move themselves out, so that at the very end, and this is a key part, there's going to be an equal distribution of these molecules or particles throughout this matrix, or throughout the water. So even though if we see this as really a kind of a dark red, over time that dark red gets lighter and lighter and lighter, because it's going to have an equal distribution of the particles throughout the water. So let's take a look at what osmosis is all about. Now, osmosis is a little different because there's a membrane involved, and a very, not any old membrane, it's a semi-permeable membrane, which means it has a series of small microscopic holes will allow certain particles in, and of course keep other ones kind of behind it, because of the size of those pores, or the pore spaces in there. Now, in diffusion, there is no membrane, and that's a clear difference between the two that we have to look at. So this is kind of a very popular illustration. You have these U-tubes here, and in the middle of this U-tube right here is that semi-permeable membrane. Notice these little holes, microscopic holes in here. And in this case right here, what you're going to see is there's going to be water on each side of them, but also inside, dissolved in the water, is the solute. In this particular case, it's sugar. And you can see on one side it has more sugar, or solute, on one side. Like on this side, right here, there's more water in here. So this is a higher concentration of water particles, because the sugar right here is not quite as concentrated as on this side over here. On the right-hand side, you can see there's a lot more sugar molecules. And sugar molecules are, especially if they're something other than just glucose, it's going to be like a polysaccharide, like for instance a starch. That's going to be a lot larger. And because it's so large, it's not able to fit through these holes. So this, on this side, the left side, has less solute and stuff that's actually the solids that are dissolved into the solvent, and this has more solute. Now, another way to look at it, because it has more solutes, or solid things that are dissolved into the solvent, is what we call hypertonic. Hyper means, of course, it has more pressure on it, so there's more particles in here. Whereas the hypotonic means there's less particles on this side. Now, because this semi-permeable membrane in here, it's going to kind of segregate the size of the particles. So because these are large particles, the sugar's large, the water particles are much smaller. Because if we take a look at, here's this maybe a little particle of H2O. It's not very big. But when we take a look at a glucose molecule, there are 6 carbons in here. It's a much larger molecule. It's not able to fit through here. So what happens is, because these can't fit through here, there's going to be many more, or a greater concentration of water on this side than on this side. So in this particular process, what the water wants to do is it wants to kind of level it out. It wants to kind of have the same amount of water particles on each side. So what's going to happen is the water molecules are going to come across this membrane and fill in this right side. So when it does that, it's going to look like this. Once the water starts to level off and it hits the same amount of concentration of water molecules, that's the key, because the semi-permeable membrane prevents the solutes from going back and forth, you're going to see a difference in a gradient in the height of the water in each of these tubes. Now this is just an illustration, and I kind of wanted to make our own, and I tried and it wasn't very successful, but there was a company called Pasco Scientific that makes an apparatus just like this so that we could actually do this demonstration, and that's what we did, and that's what I'd like to show you next. So what we did in class, before we do that, let's take a look at the difference between the two. When we look at diffusion and osmosis, you're going to look at this comparison. Diffusion, first of all, wants to move all of the particles to equalize the concentration. So because there's no membrane in the center, the particles are free to move throughout the matrix, and it wants to go from a higher concentration to a lower concentration, and the end result is going to be an equal distribution of all of those particles. But there's no semi-permeable membrane involved, none. So when we look at osmosis, on the other hand, now remember, only the solvent particles are going to move in osmosis, primarily because of the semi-permeable membrane that's not present in diffusion. And the solutes really aren't going to be moving. So the movement is actually through the semi-permeable membrane. It's kind of the limiting factor. So the solvents are going to move from high concentration to low concentration, but the solutes kind of stay in one place. So there'll be an unequal distribution. Now, both of them have to do with the movement of particles. That's a similarity. And they want to equalize the concentration of the two solutions. They really do, but because of the semi-permeable membrane, that prevents that from happening. So let's take a look at the lab that we put together. And we use these devices. The initial pressure is the first thing that we had to look at. So instead of just looking at the height of the water in each of the tubes, we had some sensors attached to the very tops of the tubes so that we could measure the amount of pressure. So not only will we be measuring the volume or the mLs, but we're also going to measure the pressure to see that difference between the two. What's the relationship between the two of them? So what we did, let me see if I get a little closer. Whoops, let's go back one. In this particular case, if you take a look at this, we have right here two sensors hooked up to what we call a data logger, which hooked up also to the computer. These are pressure sensors, and they're attached to the very top right here of these caps. And they're basically caps that we can screw down and cause a very good seal in here so we can capture the amount of pressure. Now, if you look right here, they're filled up to basically the same across the two. This one right here is sugar water, which is basically dissolved the same amount of a portion of Caro syrup into water. Then over here we're just plain old tap water, and I colored it blue just so we can see some differences between them. So this is just regular H2O, and there's water and sugar in here, and of course just water there. So once we get that all hooked up, let's go back one more here. There we go. All right. Now notice the caps are sealed. Notice these caps are sealed, and they're hooked up to the data loggers. We put 40 mLs of water, and we also put 40 mLs of sugar water, and then this happens to be distilled water right here, and I put dye in it. So we have 40 and 40. And you can see there just right at the same mark. Now right down here, right between the two tubes, is a membrane. and it is very much like a dialysis bag. It has small little bitty microscopic holes, so there is a connection between the two of them. And at time 0, this is what it looks like. All right, so everybody can see that. That's time 0. Everything pretty much looks the same. So let's go to the next picture. Now, the other thing that we measured is also the pressure. And the pressure is really important because that will tell us kind of where everything stands at this particular point in time. Now, if we look at this, we can see that still at 40 mLs, 40 mLs. The pressure is slightly different. And the sugar side, which is actually over here, we have 106.6 kilopascals, and over here we have 106.25 kilopascals right there. So that's what it starts with. So those are our initial readings. And now what's next is we're going to take a look at the 10th hour. And in the 10th hour, you can see some remarkable changes going on. If you take a look at the pressure itself on the sugar water, notice it's slightly higher than, of course, the distilled water. Now notice it's starting to crawl up the tube, and as it crawls up, it's putting, of course, pressure on the air molecules, and those air molecules are kind of bouncing up here, causing a different pressure reading, which means it's a little bit higher pressure than over here in the water, which means there is movement of water molecules from here across the membrane and into here, right into the water. So now the sugar water is getting less and less concentrated in terms of the amount of sugar molecules to water molecules. It's becoming a little less hypertonic. So that's what we have right here. Notice, if you will, now the volume of water is starting to drop, which means the pressure is starting to drop because there seems to be now more space for the air molecules to bounce around, and because of that, there's less pressure. So what happens here, let's go to right there. If we take a look at here now, we can now look at the level of the volume. And on this one, on the sugar water, it's 41.5, and over here is 37.5. So again, the water is starting to move downward, and this is being filled up because now osmosis is taking over. That semi-permeable membrane is not letting the sugar molecules get through the membrane, but it's allowing the smaller water molecules to sift through there, and then, of course, fill up the left side where the sugar water is, and making it less dense in terms of the sugar molecules to the water molecules. Now, we're going to go 24 hours later. I called it the big finish, and let's see what we've got. Here is what happened at the end of 24 hours. We let it go 24 hours. And by the way, we let a few of them go a little bit longer, and it still started to push it up because we still couldn't get that even pressure gradient, so that draw to even out the pressures was still very strong, just because of the amount of water molecules in each side. In this one, we can see that we had a loss. We started with basically currently right now the pressure was 91.72 kilopascals. That's in a loss, and it lost in 24 hours of 14.5 kilopascals. On the sugar side of things, though, things were a little bit different. In this particular point, when we were all finished, is 125.58. That was a gain of 18.93 kilocalories. So you can see that it definitely had come up quite a bit. Not quite even. Notice, if you think about it, it should have been even. If this lost 14, that should have gained 14. But that isn't totally, in the real world, it doesn't always totally work out nice and clean. So this one didn't either. But again, notice where they stopped, where the volume was, and we'll get to that one in just a second. So let's go over to the volume side. And then you can see another big difference. Here on the water side, there was a loss of about 6 mLs. And over here, there was a gain of 5 mLs. So you can definitely see that there was a difference. The way you can explain it is through osmosis. It allowed the water to be able to move through the semi-permeable membrane from the water side to the sugar side. And what caused that movement? The process of diffusion says, hey, look, there is less water over here. We have greater water on our side. So that process allowed for the water in the distilled side to go through the membrane and, of course, push the water up in the tube on the sugar water side. All right. So let's get back to the very finish. All right, so what's the difference between diffusion osmosis? Diffusion is basically the movement from a greater concentration to a lower concentration, but it doesn't have a membrane. Its goal is to equally diffuse or to place all the particles equally throughout, so there's an even distribution. Whereas osmosis works within a semi-permeable membrane, which only lets some things in and keep others out, and it usually is an unequal distribution of particles. But again, in osmosis, it has the membrane, diffusion doesn't. I hope that's helped, and when we do the evaluation in our lab, I hope you use this to help answer some of your questions. So thanks. Bye.